Hello and welcome to the ZOOMinar. Today we have two speakers, Dr. Guido Pintacuda from Lyon University and Dr. Jinghui Luo from Paul Scherrer Institute located in Villingen, Switzerland. Let me briefly introduce the first speaker, Dr. Guido Pintacuda. Guido obtained bachelor's and PhD degrees in chemistry from the University of Pisa, Italy. After working at the Karolinska Institute in Stockholm and at the Australian National University in Canberra with Professor Gottfried Wotting, he moved to University of Lyon in 2005 as a CNRS researcher. In 2009, he became the research director at Lyon in the NMR Center. Since 2018, he has been the head of the Lyon High Field NMR Center, where he develops NMR spectroscopy applied to study exciting biological problems. And he leads the research team and the development of NMR methods at high magnetic field. He was awarded the Raymond Beverly Sackler Prize in Physical Sciences in 2017. Since then he has been working on developing methods and applying to study high resolution structures. Guido, welcome and get started when you're ready.
Yep. So thanks, Rams, and thanks to the organizers for the kind invitation and the nice introduction. So I'd like to talk to you today about, so as you said, Rams, to conformational dynamics by NMR in crystal. So it's a weird titles and a weird subject because so the main topic of the talk is solid state NMR. Solid state NMR, and when you see, I mean, figures like that, so this geography of the different protein states that are present in the complex folding pathways of biological systems, when you think of I fields and NMR, solid state NMR, you invariably associate them to the determination of level structure and dynamics and properties of fibrillar space, the species. Now, today I would like to sort of open for you a new window and tell you something about the unique potential of solid state NMR to study the same network of interactions, but from a different point of view, studying them from a crystal point of view. Well, first, actually, I'll divide my talk in two parts. First, I can't help telling you something on the power of solid state NMR of fibrils and update you on the latest sort of progress in this area. So you all know that solid state NMR has this unique capacity to determine atomic level structures of fibrils. There are a number of examples depicted here on the left, and Rob Tico, Bob Griffin, Beat Meyer, and many others have shown this. So I've led this pioneering determination, and there are established protocols to transform NMR spectra acquired in the solid state into well-defined structure and structure ensembles. So I cite here a review that I co-authored with Antoine Loquet's laboratory and David Abenstein a few years ago on this subject. Now, the news in this area is that the solid state NMR field has undergone a revolution over the last few years. Revolution based on the fact that it's possible based on the development of rotors, so the sample holders used in NMR, which are smaller and smaller and capable of rotating, spinning the sample around the magic angle faster and faster. So the commercial rotors produced by Bruker, exemplified here, now go up to 111,000 Earths. So it's more than 100 kilohertz in very small 0.7 millimeter rotor, which entails a reduction in the sample volume required for the NMR experiments, but also open the possibility to study solid state spectra based on the detection of proton resonances. Protons are the most abundant in proteins and have the largest geomagnetic ratio. So associated to that, there is a huge sensitivity gain. So you can simplify the years this transition from the traditional solid state NMR spectra based on carbon detection at slow spinning rates in large rotors with large sample quantities. This is what you may be familiar with. So solid state NMR, something which really takes a lot of samples, a lot of time to acquire spectra, with a transition to deuterated proteins. So proteins where the proton content is diluted by deuteration and reprotonation of the amide sites, for example, in the 60 kilohertz regimes, which was the state of the art till a few years ago, and since 2015, 2016. Now it's possible to work on over 100 kilohertz in very small rotors on and run resolved proton detected spectra on fully protonated proteins. So you can see here this visually this transition. On the left, the proton-nitrogen correlation spectrum, you can get a slow spinning speed, useless. And on the right, the proton-nitrogen two-dimensional spectrum that you can get in 10 minutes with 0.5 milligram of sample over 100 kilohertz. So this is a revolution because it opens the way to the rapid fingerprinting of biological solids and rapid structural analysis at atomic resolution. So we published a proof of principle study on PNAS in 2016, studying some microcrystalline proteins, a smaller one and very larger one. This opens a lot of avenues, but is this relevant for the study of amyloid fibrils? So amyloid fibrils may be intrinsically heterogeneous. They are the presence of many polymorphs, could be more dynamical. This methodology may not work for these kind of samples. And actually in the community, there was quite some doubts that this technique, the possibilities of this technique could be extended to more heterogeneous sample. And actually, I would like to show you some selected data from two collaborative studies, one with the laboratory of Birgit Abelstein and Antoine Loquet in Bordeaux, and Sven Soap, that we published last year, and one which is just out this year, but started many years ago with the group of Bob Griffin at MIT. So these are two studies on amyloid fibrils, and as you can see here, on the left is Bob's A-beta42. It's a fully protonated sample of fibrils from A-beta42. On the right is the spectra from the ELF. So it's an homologue of the ETS, so this prion protein from the fungus Podospora anserina, that has been a model for the development of solid-state NMR for many years. And as you can see in both cases, you can have very nicely resolved proton-nitrogen and proton-carbon fingerprints, which can be acquired again rapidly and have a very, very, very incredible resolution. On the gigahertz, this has 200 or less than 200 resolution. These lines are narrower, are broader than the microcrystalline samples that I've shown you earlier. But nonetheless, this provide a window for rapid resonance assignment. And here we have developed over the years strategies to similar to solution to the solution and the NMR counterpart to link this proton-nitrogen spectra to the carbon frequency in 3D spectra, carbon, which could be carbon alpha, carbon beta, or carbon, the same residue, the residue just before, allowing therefore to link them over the sequentially over the backbone. Or similarly, we can use this two-dimensional proton-carbon correlation to open a third dimension, linking all the spins, the carbon on the proton spins of the side chains, and therefore getting resonance assignment overall of the protein. Now, these spectra, they may look very time-consuming and very complex. Actually, they are extremely rapid, extremely simple and rapid to set up and to acquire. So even on a non-crystalline amyloid fibrils, they can be acquired in less than a week, maybe a couple of days, 3 days, and provide a set of data which is extremely self-consistent and can be actually analyzed automatically without even manual interventions. Once the peaks are picked, there are algorithms. We use very often Flyer from Peter Gunther, who is very easy to customize with respect to a given input to get automatically the assignment of a resonance. So this is, I'm telling you this because a major roadblock which has hampered the use of solid-state NMR in structural biology has today been removed. So all this can be really done and streamlined very, very rapidly. So we have even come to the point that all these shifts can be acquired in hyperdimensional experiments, which are very impractical to run in solution, like 5D experiments, so hypercubes. But you can actually even acquire as projection spectroscopy, the APSI. So these are concepts which were introduced in solution NMR, but then abandoned because actually by solution NMR it's very difficult to study very large molecules that require this complex setup. And finally here, this is an Angevante paper where we demonstrated this with this approach a few years ago. You can collect data in 2-3 days and then paste into topspin. So run this to a APSI topspin module, get the peak list, feed the Flyer software, and automatically, without even spectral analysis, we can obtain the resonance assignment of this species. The species which can be microcrystalline, but also fibrillar. And so this is extremely powerful because once you have the assignment of the spectra, well, you can use them to sample, to monitor, to probe very efficiently proton-proton proximities. And for example, in a fibril, you can probe the network of proximities that define the amyloid core of the structural core of the fiber. So this is an example of the CHH or NHH or the CCH spectra, so 3D, complex 3D spectra, but can be acquired only in a day or two on these samples in a fully protonated form, and therefore facilitate the calculation of a structure. Here in this paper we just highlight how the data can be collected, and in the work on LF, actually we carried out we carried out the full structural determination. So this network of proximities can be translated in a structural bundle. And here, Benjamin Barbieux in Institut Pasteur was essential to program the structural calculation. So what we here, I show you this because together with the acquisition of the distances, we also engineer some particular labeling schemes allowing to distinguish between the inter and intramolecular contacts which define the packing of the monomers in this fibrillar architecture. Now, this is nice because this accelerates significantly the capacity of solid-state NMR to investigate structure of fibrils. Now, of course, at the same time, other techniques have also progressed, and of course, we all know that cryo-EM spectroscopy has become one of the very solid alternatives of solid-state NMR for the determination of the species. Nonetheless, the capacity of solid-state NMR to acquire this information very rapidly is still carries still a great potential, and this acceleration will be very important in the future. Now, these are the news, or at least the news from my point of view, about the solid-state NMR and amyloid fibrils. But what I promised you from the beginning is to tell you something else, to tell you what solid-state NMR can tell about dynamics of proteins in crystals. Now, crystals or microcrystals or nanocrystals actually have been the foundations of the development of solid-state NMR over the last 20 years. So there is a seminal paper by Martin and Zil in 2003, but many other groups, Anna Dermot, Bert Meyer, Anja Brackmann, at the time we're working on similar ideas at the time, and they realized that actually large crystals or small crystals, so what crystallographers call very often a precipitate or a powder, may have a local order, and this local order, which is X-ray crystal, the determination by X-ray crystallography based on long range order, they require large crystals. By solid-state NMR, the quality of the spectra acquired on polycrystalline, so large crystal, a set of large crystals, on the nanocrystalline sample, where the size of the crystals is much smaller, actually have no difference. So the NMR quality, spectral quality, is only determined by the local order, so is independent of the size of the crystal. And crystals are very easy, nanocrystals are very easy to obtain, so precipitate, just by adding a precipitant, PEG, MPD, other composition, which can be screened very rapidly. Then you have this powder, what you can collect in pipette tips. In the early days, now there are sophisticated tools like funnels, where you can collect the crystals, large quantities of crystals, and then centrifuge directly in the NMR rotor, which then you place in the magnetic field and run your experiments. Now, crystal samples or microcrystalline samples, therefore, are easier to prepare, and have been the playground on which most of the methods for solid-state NMR have been developed in many years, but have been considered in the field only just model samples or exercise samples. The real things, the real targets, the real objectives being fibrils, being membrane proteins, being other non-crystalline samples. What I would like to tell you today is that it is very interesting actually to study microcrystalline samples themselves. So many say, well, why to study a sample if you can also run X-ray crystallography on that sample? Well, first of all, as we could see, as we saw, not always a microcrystalline sample is associated to a large crystal on which you can run X-ray crystallography. But even if a larger crystal can be produced and X-ray data are available, solid-state NMR provides something different. So beside the structure or the structural characterization of the sample, solid-state NMR carries unique potential to study dynamics. And so these slides, it's a figure from a review that we wrote with a student last year in 2021, actually shows that over the last 20 years, on the left, you can see there are a number of technological progress from new probes, new magic angle spinning probes, new spectrometer, new fields, DMP apparatus, the capacity to run NMR at cryogenic temperature under magic angle spinning. So a lot of technological progress. And in parallel, on the right, this has produced a number of techniques, a variety of techniques, from the older one in dark blue to the newer one in red. So it's a gradient of developments. And these techniques are, so these are new NMR experiments which can probe dynamics in a sample, dynamics ranging on a large variety of time scales from picoseconds, nano, micro, milliseconds. And you can see here the recoupling of anisotropic interactions that is more associated to the faster time scales, spin relaxation, large window, relaxation dispersion techniques, a new set of developments which are more recent and are capable of probing slower motions. Flash freeze trapping, chlorine exchange. So today I'll focus actually on 3 experiments which can be evaluated once you have a two-dimensional correlation on the left. For each signal, you can measure some parameters, some relaxation parameters. So these are for what I will discuss today: the nitrogen R1, the longitudinal relaxation, nitrogen R1 rho, the transversal longitudinal relaxation during a spin lock, and the relaxation dispersion. So the variation of the R1 rho with while the spin lock field is changing during an experiment. And with these 3 different experiments, you don't need to look at the details, or you can check out the references of the first time these experiments were used here, which are indicated here. But what you need to retain here is that each of these experiments is capable to probe. It's very sensitive to a different time scale. Nitrogen R1 nanosecond, R1 rho microseconds, relaxation dispersion micro to millisecond. Now, the study of dynamics in crystals has accompanied the development of solid-state NMR. There is a progression of studies on model systems from 2005, Nicolas Giraud and Lyndon Emsley in Lyon, Chevelko, Reif, Oshkinet in 2009, Paul Shander, Jax 2012, Lewandowski, Martin Blackledge, Megan Else, and Lyndon Emsley, Science 2015. So there are a variety of experiments which were run on small model systems. But from my point of view, I think that there is a step change here on the use of this spectroscopy for the study of dynamics, with the application on larger molecules which contain some functional dynamics. And I present here, I show you 2 examples: the human carbonic anhydrase from the Linzer's lab, and the human copper zinc superoxide dismutase, which has been a PET molecule in Lyon for many years, but that has been the object of a collaboration between Emsley, my group, and Ivano Bertini's group laboratory in Florence. What is interesting here is that these large molecules are enzymes, and if you study them in the crystals, actually the crystals are active. So the substrates are small molecules, so carbonic acids, superoxide ions, which can diffuse through the lattice of the crystal, and the molecule is the enzyme is functional also in the crystal form, in the solid state. Therefore, it is interesting. It becomes very pertinent to study dynamics and compare it with their performance. So the first point is that here, superoxide dismutase, 32 kilodalton homodimer. As I said, this is a long-standing collaboration between Lyon. I indicate Lyndon as Lyon researcher because he was still in Lyon when we started, and in the Florence group. It's very difficult to say something new on superoxide dismutase. It's a very well studied molecule, a lot of literature there. This is a very efficient enzyme which contains zinc ion and the copper ion, which is copper 1 or copper 2. And but actually, the reason why this molecule is very well studied is that it's associated to amyotrophic lateral sclerosis. So it produces some deposits, some aggregates, and there are a number of mutations that accelerate the production of this disease. There are mutations around the metal center, the metal binding sites, or mutation at the interface of the dimer. Now, mutation at the interface of the dimer is very clear. It's very clear what effect it does. It destabilizes the dimer and therefore accelerates aggregation. It's less clear how a mutation on the active site next to a metal center can produce a destabilization of the molecule. Actually, this is what we investigated by solid-state NMR. This is the proton nitrogen two-dimensional correlation of a molecule with a copper zinc and the dimer without copper, or with only the zinc ion. Plus, minus copper, the spectra are extremely similar, very small chemical shape differences. And nonetheless, so there are no structural evidences evidenced by X-ray crystallography, but nonetheless, we do measure dynamics, and we do see differences. Now, if I tell you, if you add a metal ion, what's the effect on dynamics? I bet that most of you, all of you would say, well, the molecule becomes more rigid. Actually, if we look at the nitrogen R1, so the fast dynamics, we find the opposite. The molecule with copper becomes more dynamics, and the motion happens in particular areas of the protein, loop 4, 6, and 7, which is the loops flanking the channel leading to the active site. So what happens is that actually copper binding selectively activates motions that help the diffusion of substrate to the active site nanosecond. Dynamics is the time scale associated to diffusion of the substrate in the active channel. But then, if we look at another time scale of dynamics, we look at the relaxation dispersion, or R1 raw relaxation dispersion, slow dynamics. We observe the opposite. We observe a completely different effects. We see that the active site is unchanged. Dynamic is not altered at the active site, but dynamic is increased in the absence of copper at the interface of the dimer. So here, what we find is the opposite. So without copper, there is the microsecond, millisecond exchange, which decreases the stability of the dimer and perturbs the interface of this molecule. And what we could also find here is that there is an histidine which bridges the active site and the interface, which anchors the different regions and transfer dynamical effects, that distributes dynamical effects from the fast time scale at the active site to redistributes it to the slower time scale at the interface of the molecule. So this is intriguing because it's an example that shows that dynamics in crystals can provide evidence of the function of the molecule, but also on the dysfunction of the molecule. And I think that all this becomes a little bit more clear in a second example, which is a collaboration with Stefano Ricagno in Milan. So Beta2 Microglobulin, so another well-studied molecule associated to amyloidosis. But what we studied here actually was a particular form, a particular mutation of Beta2M, which increases tremendously the aggregation propensity. It is associated to a disease found in some families of subjects in Europe, and which dramatically transforms the aggregation propensity of the molecule. You can see here the wild type has a no fibrillation, physiological pH, physiological conditions, and extremely fast fibrillization in the D76 end variant. Nonetheless, crystallography is blind on that, because if you study the wild type molecule and the variant, the molecules are absolutely identical, very small RMSD. And so they are blind. Nonetheless, this 76 position seems to be particularly neuralgic in the properties of the molecule. Now, what we started our crystallographers, our collaborators started looking at variations of the amino acid type at these 76 positions. You can get crystal structures which are absolutely identical, apart from different network of hydrogen bonds in the loop carrying the mutation. Nonetheless, every time you perturb the D amino acid, you get a reduction in the stability of the protein and an increase in the fibrillization propensity. So X-ray crystallography is clearly not the right technique to study this phenomenon itself. So this is why we turned to NMR. You cannot do NMR studies in solution because the molecule, the D76 molecule, the variant, fibrillizes during the NMR determination. So by studying crystals, we have a further advantage that the crystal lattice protects the molecule one from each other and sort of shields them from the interaction and yields a sample where we can observe the intrinsic properties of an isolated molecule. Nonetheless, if we compare the wild type and the mutated spectrum of the molecule here on the left, spectra are extremely similar. And the chemical shift changes are very small in both cases. Here, the red and the blue are superimposed. You cannot even really see the two curves, distinguish the two curves from on top of each other. Actually, there are extremely small changes localized on some loops, but are extremely negligible. We went on and determined nitrogen R1 raw. This is a mistake here, out of which you can analyze, which can be analyzed in terms of motional amplitudes and time scale of motion in the fast time scale window. And dynamics is conserved in both cases. These data are consistent with an extremely rigid molecule, which is the case. I mean, can be expected on a small globular protein in a crystal. Nonetheless, there are some differences already quite intriguing, in particular in this loop carrying out the mutation. These profiles and the extracted order parameters can be reproduced very well by molecular dynamic simulations of isolated molecules, which proves, again, that what we are looking at are really properties and motion of an isolated molecule, but the effect of a crystal lattice is not having, at least on this time scale of motion, a big effect on the dynamic. What is extraordinary, I think, is that when we looked at the nitrogen relaxation dispersion, so much lower time scale, we could see many residues having a very flat profile, nothing relevant happening. But if we look at the wild type, well, we can see that there are some amino acids which display dispersion. Dispersion means that have indication of a slow conformational change in the micro to millisecond window, and they were centered on the loop carrying a proline 32. Of course, so it could be a trivial proline isomerization happening there. This is on the wild type. The rest of the molecule has no effect. But when we look at the mutant, we see that there are exactly the same profiles in the loop carrying the proline 32. So these dynamics, maybe uninteresting dynamics, is conserved. But in addition to that, we can see relaxation dispersion happening in sites where the wild type had flat profiles. And there are all the residues here of the loop carrying the mutation and some residues of the flanking beta strand. So the terminal part of the first beta strand, the A strand, the beta strand actually closing the beta barrel on the N-terminal side. Now, these changes are extremely intriguing, and it became even more intriguing when we teamed up with Carlo Camilloni, who ran Replica average metadynamic simulation, which based on the NMR data allowed to extend the time limits of the classical MD simulation and provide structures or structural ensembles, so calculate structures and group them based on their free energy profiles, clustered according to some internal variables, the side chain Rothermer distribution and the antiparallel beta structure content. So if you analyze the results of these simulations in this way, structures cluster for both the wild type and the mutant according to two bundles. At first bundle, which in both cases as very similar, larger populations and very similar structure as compared to the crystal structure. And then two bundles of which correspond to excited states, which are then present in the crystal, which have a lower population and contain, so have a structure which deviates from the crystal structure, deviates only a little in the case of wild type, deviates substantially in the case of a point mutant. And for each of them, it's possible to calculate the content of beta strands, which is significantly lower, in particular for this excited state or excited state bundle of the mutated part. So the excited state bundle, which therefore has a reduced content of beta strands. So some of beta strands are partially perturbed or unfolded, and correspondingly becomes less soluble. In particular, free regions become more prone to aggregation. And then based on that is possible, also based on these figures that Carlo Camilloni produced with the metadynamics, is possible to look at interactions, pairwise interactions between different atoms, and reconstruct a network of contacts of salt bridges where the D76 residues is central. And if you look at the N mutation, the network is perturbed, weakened, with some weakened interaction, most of them, in most of the cases, or strengthened interaction upon mutation. And it's possible, therefore, to follow the way in which D76 is a key linker between between the loop carrying the amino acids and some beta strands in different locations of the protein. So we could, based on that, probe the validity of this network by mutating the arginine 97 into a tryptophan and producing a destabilization of the protein. But it's also possible, based on this analysis, to recalculate the non-bonded energy for all the mutants that we had in the loop and correlate it with the stability, and is surprisingly, incredibly providing a linear correlation. Now, and I'm just finishing in a minute, is all this real or not? Well, it's a nice story, it's a nice dream, but does this have any relevance with respect to the fibrillization of the molecule? Well, you can see here that we can now study also the fibrils of the D76N, Beta2M. You can get spectra, you can assign them with the spectra that I described early on. And based on the assignment, you can determine at least very rapidly the secondary structure. So this is the profile of in black are all the atoms which have been assigned, and the nuclei which have been, for which chemical shift has been assigned, defining very clearly a rigid core from residue 20 to residue 80, out of which, with a Talos, which is a classical solid-state NMR tool, we can predict the secondary structure. And this clearly yields 5 regions with beta strands, which can now be compared with the native fold in the crystalline state. This is the fibril, but can more interestingly compared with excited state ensemble that Carlo calculated based on the NMR data in crystals. And you can see there is a gradient of secondary structure elements upon fibril formation. And the regions where we did detect conformational changes in crystals are actually the ones where the beta strands of the native fold disappear in the fibril or are modified. For example, in this case, the C and D strand disappear in the excited state and reappear as a longer stretch in different regions. And also here, the D76 loop, where we have the largest dynamical feature in the mutant. Well, this is a region which acquires a secondary structure characteristic as a beta strand in the fiber. Therefore, the destabilization of the native structures, what we can spy by solid-state NMR, may provide the hint for a path, for a mechanism for the fibril formation for the formation of the new fibrillar architecture. And you can see all this sketched here: crystal structures where we have ground states, which are very similar, and excited states, which in one case in the wild type show a protected beta strand core, and then a more exposed core in the D76N, allowing an easier formation of fibrils. And I finish here thanking, well, all the people that over the years in Lyon have contributed to the development of solicited NMR spectroscopy, proton detection, and the study of dynamics in crystals in particular. Well, Tanguy, Marta have been key players for the examples that I mentioned here. Thanks to a large number of collaborators from Europe to the US at Bruker in Florence. And just the very final thing, we are working as a large-scale facility, open at a national and international level through two funding schemes: the Infranalytics run by by CNRS, and INEX Discovery run by the European Community. So if you are interested to try to look at dynamics in crystals and many other examples, many other experiments with solicited NMR, please apply, and we will provide NMR time and assistance for your project.
Thank you very much, Guido, for a great talk. Lovely research and a great technique. Audience, please post your question in the Q&A folder. It will be addressed. I already see one question from Rob Tico. Let me see if we can get that.
Yeah, so I can see the...
Yeah, a change in R1 can be caused by a change in timescale of motion or amplitude motion, motional amplitude, or both. Can you disentangle and quantify these effects in your measurements on SOD?
Yeah, so it is possible to run more sophisticated analysis than what I presented. So I presented actually finally I just presented the raw data, which by themselves are an indication of a redistribution, as I said, of motions over the different time scales. Nonetheless, by taking all the data together, and we have also data at multiple fields, and so R1 raw, carbon R1 raw also at the multiple fields, is possible to do a more sophisticated analysis and introduce a emotional model. For example, we run a Gaussian amplitude fluctuation or simple model free, or we run also extended model free by adding also other determination of dipolar couplings. And in this case, it's possible to indeed disentangle this effect. I think for simplicity, the data themselves without the analysis sometimes are very talkative, and I prefer to present the data that way. But yes, this is possible, and it's very exciting because the NMR again is the only technique that can disentangle motional time scales, amplitudes on over so many at atomic level.
Thank you, Guido. So I have a question for you. You showed so many ways to measure dynamics, so many different NMR techniques. Why can't you use a simple NOE-based NOESY-like experiment? I didn't see any of those.
Well, NOE, NOE is yeah. So NOE is one of the effects of the signature of dynamical processes, right. And which is is not actually the easiest to measure. So NOE by solution NMR, you're always most of the people think of proton NOE. So protons in solids are subject to a very even at the fastest rates are subject to a lot of residual coherent contributions, and cannot be easily used for that. So you would need to use nitrogen.
Mhm.
So proton nitrogen, proton carbon, and these are more experiments which are more tricky to acquire, and sometimes in fully protonated proteins are polluted by exchange contributions. So contributions, so a fast proton-proton exchange on exchangeable sites, and then the transfer by another. So the experiments are not so clear, so clean often to analyze, but in many cases, well, in some cases, I think that
Definitely, in the
also
case of
be,
protons, it might
yep,
be very difficult due to coherent
yep,
dipole-dipole coupling. So
yep.
there's a question from Jinghui Luo. A very nice talk and interesting technique. You mentioned copper 1 in SOD. How did you determine copper 1 in SOD? Why not copper 2?
Yeah, so both molecules can be studied, and we have done that. We keep here the copper in a copper 1 state, in a reduced state, just to separate clearly the effects of dynamics by the effect of paramagnetism. Copper 2 is paramagnetic as unpaired electron, so the D9 ion, which has dramatic effects on the relaxation rate. On the nitrogen, the effect is minor, but still non-negligible, and also bleaches out many of the rest of the signals from the spectrum. These are extremely important effects which can be used for the structural characterization, structure and functional characterization of the molecule. But here, really, we want to disentangle the two effects and just use a single diamagnetic oxidation state.
So for the benefit of non-NMR people in the audience, I would like to ask this question that, can you tell us how much time versus how much sample you need to take a best candidate, let's say homogeneous fiber, you have it in abundant quantity. So how much you need, how much time of measurement you need to completely solve the structure for an amyloid fiber? Can you give some idea for the audience? Yeah.
Well, today, so it's very difficult to generalize because there is only one sample. So this the LF, which we worked on together with Antoine Loquet, Antoine Loquet's group, which we did run the full structural determination on a fibrilla sample. So it's very difficult to extend or to generalize the.
It's okay. Give some parameters. How much sample? How
Yeah.
much
So the sample was half of a milligram or slightly less. So you don't have a full control of the quantities, but it's
how much measurement time?
Measurement time is for the structure for the resonance assignment was about a week. Was the first time we ran the experiment, so this could be could be possibly reduced today, and less than a week for the acquisition of the restraints, so the proton-proton proximities. In this particular case of LF, we also ran a second sample with a mixed labeling to really disentangle the intermolecular contributions. But otherwise, the samples I just mentioned, it's a sample with full protonation and just nitrogen and the carbon labeling.
So you mentioned about full protonation. You also mentioned about proton-proton distance measurement. What are the challenges you see in the case of protons, because you have multiple spin systems coupled to each other? How clean and how easy, even in the best sample scenario?
Well,
and a very fast, let's say 150 kilohertz, even then, it's still difficult.
Yeah, I think that the proton-proton proximities are extremely easy to determine in these conditions for beta strands, for beta barrels. And well, it was extremely easy to detect the hydrogen bond pattern. I mean, the bonding pattern it is very easy to determine hydrogen bonding patterns in fibrils And if you look at most of the structures, the membrane protein structures, which have been determined so far up to date by solid-state NMR with proton detections, they are all beta viral. So in this class of systems, we are quite doing quite well.
Thank you very much, Guido. Just stick around. We'll have more discussion at the end of the seminar session. So, Jinghui, can you share your screen?
Yes.
Okay, our next speaker is Dr. Jinghui Luo. Jinghui received bachelor's degree in engineering from Zhejiang University in China, master's in applied biotechnology from Uppsala University, Sweden, and a PhD in biophysics from Leiden University. After completing postdoctoral research in Hagen Bailey's lab at Oxford University, joined the Paul Scherrer Institute in 2017. His main research focus is on amyloid aggregation, and today is going to talk to us about transmembrane protein nanopore engineering for amyloid protein studies. Jinghui, take it away.
Thank you very much, Rams, for a very nice introduction, and also for the invitation from committee. So good afternoon, good evening, good morning, everyone from everywhere. So today I'm going to talk about transmembrane protein nanopore engineering for studying amyloid proteins, which is one of our research lines. And the other research line I would briefly introduce, which mainly we access to large research infrastructures, as you can see here, like Swiss light source, neutron source, and the Swiss sphere here. So in this talk, I have two parts. The first part I would introduce, try to engineer pore-forming protein like alpha hemolysin for structure oligomer structure determination and also antibody screening. And the second part I would use alpha hemolysin nanopore.
Your slide is not forwarding. Can you go to the presentation mode?
Okay. Yes, I think this is a presentation mode.
No, I don't see that. The slides are not moving, so somebody else also say the same thing. So did you share your computer, or you shared only the file? So maybe you want to.
I will share again.
Do it, yeah.
So can you see my screen now?
Now working well now. Go ahead.
Okay, that's great. Yeah, so in second part, I will introduce use alpha hemolysin nanopore to detect amyloid proteins. So, okay, so as a background, you probably already know very much. As aging, the healthy brain can become shrinked, and then you can see that lots of amyloid deposits in the brain, like amyloid beta tau protein, Alzheimer's disease, which can be scanned by PET-CT shown here. So I would like again use amyloid, Alzheimer's disease amyloid beta as an example. Amyloid beta can accumulate damaged neurons and then eventually can cause neurodegeneration disease, which probably you guys know very well. And as one of main hypotheses, the amyloid protein can interact with lipid membrane, cause leakage of cellular membranes, eventually can induce cell death. So there are a few models for the lipid leakage. For example, the amyloid protein can form toroid pore, and then it also can aggregate into beta sheets oligomer, which can insert into lipid membrane as a barrier model. So that eventually all of them can deposit as the fibers, which is the indicator for the disease. But then in reality, the most toxic one is oligomer. So we want to study oligomer, then we are able to understand the molecular mechanism of the toxicity of amyloid proteins. Unfortunately, this protein is very heterogeneous and also transient. So it's quite difficult to determine a beta oligomer structure or alpha-synuclein tau protein oligomer structure. And if we don't have a stable structure, I think it's quite difficult to develop a very conformational specific antibody against a beta oligomer antigen. So based on this research question, we would like to go to understand, determine structure, try to stabilize oligomers. So the one structure previously determined, amyloid beta, can form helping structure. So the helping can assemble together and then form kind of pore and eventually can permeabilize lipid membrane. So and the other setup in biology, you know, the toxin protein like alpha hemolysin, which has been used for DNA sequencing or RNA sequencing by using nanopore, for example, Oxford nanopore technologies. The first toxin protein, basically they used the alpha hemolysin as a model to sensing DNA or RNA. So this is very stable pore and very well-defined pore in lipid membranes. It's only heptamer structure you can see here. So there's our extracellular domain, and then see in this blue one, and the red color, this is a linker, and then you see the green one, they are helping structure transmembrane area. So inspired by this model, we would like to engineer amyloid beta helping on alpha hemolysin. To further illustrate the idea, I would like to use this model. So you can see the alpha hemolysin beta stem, and then they are linked by triangle domain, and then eventually with extracellular domain. So they can form very stable nanopore in lipid membranes, which people use for sensing, different sensing. So we found that the amyloid beta from solid state NMR structure also can form helping structure, which is very similar to the helping structure shown in alpha hemolysin stem, beta stem structure. Therefore, we replace, based on the homolog, we replace the helping structure of alpha hemolysin by using amyloid beta helping. And then we hope we can display a beta oligomer on this alpha hemolysin extracellular scaffold. And then we hope we can determine structure to understand how amyloid beta helping assembly in lipid membranes. If we know the assembly, then we probably can go to understand the lipid permeabilization. Then this will give us very useful information about the toxicity. Meanwhile, if we can stabilize those helping assembly, we probably also can develop antibodies specific for oligomers. Okay, based on the structural similarities, previously other group also show the A-beta and alpha hemolysin both protein can be detected by A11 antibody. So this shows certain functional similarity. Both can be detected by antibody, by same antibody. And we also thought because extracellular part of alpha hemolysin can block the further nucleation of amyloid beta oligomer if we stabilize, if we display a beta helping on alpha hemolysin scaffold, and we assume it will not assemble into fiber anymore. And we also assume the hybrid, the artificial A-beta oligomer probably can stay very long time as a Y-type alpha hemolysin. And then therefore we can determine structure, and we also can screen antibodies. So we tried different biochemical, biophysical methods, and we would like to confirm there's equivalence of artificial oligomer if we compare with the Y-type of A-beta oligomers. And we also want to understand the A-beta oligomer stability on the scaffold, and then as well as toxicity function on the alpha hemolysin scaffold compared with Y-type. So based on those terms we can determine structure like a beta oligomer structure on alpha hemolysin scaffold by using cryo-EM single particle analysis. Okay, the first very simple experiment we try, we engineer, we replace the beta helping of alpha hemolysin by a beta sequence. And then you can see on SDS page here. So in lane 1, this is only alpha hemolysin wild type. And in lane 2, we can see alpha hemolysin artificial A beta can form also stable only one single band A beta oligomers, artificial A beta oligomers, and then you also can see monomer as well. So if we try to further more sequences, for example, A beta 11 to 42 on replace the alpha hemolysin beta helping, and then we are also able again to see the heptamers again, and if we replace with the IPP, which also can form oligomers, you probably know it very well, and you also can see there's some different oligomers formation. I guess this because the IPP has several cysteines which can cause different oligomer formation on the scaffold. So if we try with other control sequences like A beta 11 to 28 or A beta 11 to 17, replace the beta stem of alpha hemolysin, we only can see monomer here. We are not able to see any oligomers formation. This probably can give us some idea. The A beta, the full-length A beta, the truncated version, which can form oligomers itself, and if we put on the alpha hemolysin scaffold, it still can form oligomers. If other sequence which cannot form oligomer, and then if we put on alpha hemolysin, they would not form oligomer anymore. So this can give us some kind of conclusion. A beta sequence determines the oligomerization of a beta alpha hemolysin artificial sequence, and then alpha hemolysin is an ideal sequence scaffold to oligomerize A beta peptides. So move on, we try to also use ANS fluorescence to check the hydrophobic surface formation once it forms oligomers. So as you can see, we can see lots of fluorescence intensity, emission intensity with the white type alpha hemolysin. We again also see the A beta 42 and other truncated version and IPP, both of them can form, you can observe very strong fluorescence intensity. But with a control sequence which cannot form oligomers, we are not able to see the fluorescence signals here. So yeah, so this further confirm our last conclusion. So the next step, we would like to understand how those artificial A beta oligomer, the stability, can they stay for a long time? So we did THT assay. Again, here you can see the x-axis is time, y-axis fluorescence intensity. So as you can see, white type A beta can form fibers easily after a few hours, but A beta 42 alpha hemolysin, it seems the fluorescence signal is much lower. And then further, we use the EM technique to confirm is there any fiber formation for A beta 42 alpha hemolysin hybrid construct. We are not able to see fiber formation, but we do see very clear pore formation, stable pore structure here. So this can give some idea, and the oligomer. So you can see the hybrid sequence A beta, they can form oligomers, but they will not develop into fibers. So this actually we can catch the transiency or block the transiency of the amyloid beta oligomer, and then this way would give us very useful information for structure determination, and also a very useful stable confirmation for antibody development. Yeah, and we further check the lipid permeabilization. As you can see here, we basically use single channel electric recording, use a planar lipid bilayer. For example, alpha hemorrhagin can form very stable nanopore, as you can see here. And then we also have wild type Beta42 oligomers. So the Abeta, as I also mentioned in the beginning, Beta42 is transit, not very stable. And then you can see always we can see lots of spikes, and the signal is not very stable. So we also compare with the hybrid sequence Beta42 alpha hemorrhagin, which is heptamer. Again, we see lots of spikes again here, and also not very stable signal. So we compare the conductance. The conductance for wild type alpha hemorrhagin is around the 0.8 nanosiemens, and for the wild type Beta42 oligomers, it's around 0.2 to 0.5 nanosiemens, which is equivalent to amyloid beta 40 hybrid sequence. So, so this we can learn from here, wild type Beta42 oligomers and the hybrid artificial A-beta oligomer, they display very similar channel activity, and alpha hemorrhagin will not change the channel activity once we use A-beta, engineer A-beta on the scaffold. So again, the transmembrane sequence plays an important role in determining structure and flexibility of the alpha hemorrhagin nanopore. Yeah, and we again check more biological function, and we compare different artificial oligomer Beta42 oligomer and wild type beta oligomers. We use the hemorrhatical assay, which basically we purify rabbit blood cells, and then we add a wild type alpha hemorrhagin, and then we can determine HC50 value. So which is only 50% blood rabbit cells lysis, as you can see here. And then we also added a hybrid artificial Beta42 oligomers and also the wild type A-beta oligomers. We are not able to see any toxicity of hybrid A-beta oligomer and wild type A-beta oligomers. It shows if we engineer the transmembrane part, we actually can completely change the properties of alpha hemorrhagin. So the main toxicity would be contributed by the transmembrane area. So again, we use a Western blot to detect the hybrid oligomers by A11 antibodies, which is that you can see the signal can be detected by A11 antibody, and further confirm the immune functional similarity. And then we check the cell viability of those hybrid oligomers and wild type oligomers, as you can see here. So the wild type A-beta oligomer show very similar toxicity to neuronal cells, and compare with the wild type alpha hemorrhagin, which had much higher toxicity than A-beta and hybrid A-beta sequence. So maybe this will also confirm our previous studies with hemorrhatical assay. The alpha hemorrhagin is more toxic than the other version engineered A-beta alpha hemorrhagin oligomers. Yeah, and so this further confirms they have similar functional similarities. Yeah, based on those results, we would like to further pursue structure determination. We want to determine A-beta toxic A-beta oligomer by cryo-EM. So we collected a number of data, and you can see the 2D classification here. The structure of extracellular part of alpha hemorrhagin can still see it very clearly. They are heptameric structure, and the transmembrane area, which is a beta oligomer displayed on alpha hemorrhagin, actually show a bit diffused signal. So the overall structure, we got overall nice structure, 3.3 angstrom, but only we are able to fit the density, electron density map very well with the alpha hemerizing part, the beta part is still flexible. So which actually reflect to our single channel electron recording data. We showed from there, we showed the signal is flexible from single channel recording. So so then we try to use Rosetta software to use sampling methods to fit a beta sequence into our electron density map. We are able to determine build a structure. So from here we see the structure we determined has a slightly shorter transmembrane area, and also the bigger diameter than the white alpha hemerizing. So the next step, we would like to optimize amyloid sequence, not only amyloid beta, we apparently can try with alpha synuclein, and we also can optimize our sample conditions for cryo-EM. We're also trying lipid cubic phase method to stabilize the diffused amyloid beta on the alpha hemerizing scaffold. Yeah, and the model again here also can explain this. You see the sequence, the transmembrane area is shorter than alpha hemerizing. Maybe this explains why amyloid beta on alpha hemerizing is less toxic than white alpha hemerizing. Yeah, so that's the first part I would summarize, and we develop a toxin scaffold to stabilize amyloid beta, and we are able to block the transition from oligomer to fiber, which give a very useful insight for the structure determination and also for antibody screenings here. We are already developing a few interesting polyclonal antibodies. We now we're screening monoclonal antibodies. Yeah, so that's the first part. And the second part, we would like to use, again, alpha hemerizing as a scaffold to sensing amyloid proteins, alpha synuclein, which are associated with Parkinson's disease. So I use the same figure which I showed in the beginning. So the alpha synuclein, which can also form helical structure in lipid membranes, metal ions like copper, zinc, iron, which can also regulate alpha synuclein aggregation. But it's not very clear how metal ion regulate alpha synuclein structure in the environment of lipid membranes. So the second question would be, can we develop antibody compounds which can stabilize alpha helical structure of alpha synuclein in lipid membrane? So with those two questions, we would like to use alpha hemerizing as a model, and we apply voltage here. Okay, you probably cannot see my mouse. Okay. Yeah, so we use alpha hemerizing as a model to apply voltage here. We can detect the current if there's only one pole. And then you can see here open pole here. And if we add alpha synuclein protein here, we see two different level signals. So to better check, so we can assign two different levels to the two different conformations. The alpha synuclein protein don't interact with lipid membranes. Usually it can fold it on top of nanopore. And once it interacts with lipid membranes, and we are not able to see we would see more current, then we can assign the two different conformations to the two different current levels. So from the two different levels, we are able to understand the interaction kinetics between alpha synuclein and the lipid membranes. So to further stabilize alpha synuclein on the lipid membranes, we use DNA as the leading sequence to check the whole construct along the lipid membranes. So we checked with different metal ions like alpha, like amyloid. So PCPG, which is negative charge lipid membrane with 20% negative charge lipid membranes. From the chase, what we have a kinetics. We can assign fit two different events with exponential fitting. We can get tau time, like folding time and open time. Open, it means alpha-synuclein interact with lipid membranes. And then we added metal ions to the nanopore, and we can see how different metal ions stabilize or to moderate alpha-synuclein interaction with lipid membranes. So we can compare their event half-time and also the block current from here. So the dwell time and block current can give us lots of information as well. So from blocked current folded, we can understand folded structure like alpha-synuclein metal line and the negative charge lipid membranes. We can see that the copper can form more stable structure than zinc and then iron. So and then we tried to study compounds which can stabilize the helical structure of alpha-synuclein in lipid membranes. We collaborate with Andy Hamilton from New York University. They developed a number of alpha helical mimetic compounds which can stabilize the helical structure of alpha-synuclein. And then we would like to see add these compounds to the alpha-synuclein, and can we stabilize alpha-synuclein structure, helical structure in lipid membranes? So indeed, if we compare if we only have alpha-synuclein with lipid membranes, we can see two different levels, and which is open alpha-synuclein interact with lipid membrane, and other is a chapped nanopore. So once we add the compound, we only can see the alpha-synuclein interact with lipid membranes. So we will not see any chapping events. It means that alpha-synuclein will not become a folded structure. The compounds can stabilize the helical structure of alpha-synuclein in lipid membranes. So this further confirm our setup could be very useful for drug screening for alpha-synuclein, especially if we can stabilize helical structure of alpha-synuclein in lipid membranes. So that's the second part. I mean, now we are trying to working on determine oligomer structure. We try different amyloid sequence like amyloid beta, IPP, and the alpha-synuclein, which is very challenging, but I think it's very useful, very important for the field. And so the second, we would like to develop antibodies, also synthetic nanobodies as well. And in the future, we also use the same transmembrane nanopore and engineering methods to define anti-microbial peptides structure and give better activities. And we also develop novel nanopores for sensing. So this is one of our main part, which has been developing in our group. So so the second part, I mean, which I haven't mentioned here, basically we are using large research infrastructure like a Swiss light source with a synchrotron here, and also neutron source and also Swiss field. For example, we are using soft X-ray magnetic circuitry prism to understand the magnetism of copper and the iron when it bonds to amyloid proteins. We're also using X-ray tomography, which are also available at the Swiss light source, with reconstruct 3D structure of amyloid plaques in mouse brain and also in human brain as well. So that's some other activity. Like also we in one study we use SACS to study the amyloid structure in lipid cubic phase. so which I don't have time to show you guys. So that's all of my work. Yeah, so the work has been supported by my group members, so, and also by a number of foundations. So thank you very much. If you have any question, I will be very happy to answer your questions.
Thank you very much, Jinghui, for a great talk. Audience, please post your questions in the Q&A folder. They will be addressed. There's a question in Q&A folder. You can take a look at it from Ryan J. Torres. What method was used to measure conductance?
So the conductance, we basically use a current divided by voltage, and also based on the concentration of the buffer. And the second question about the kinetics, right? Yes, we are using a single exponential method to fit the data, kinetic data.
So if binding is required for alpha hemolysin to change conformations, would you expect a double exponential?
Yeah, so we also can use double exponential to fit, but in our case, we check the data. It seems single exponential is easier for us to work.
So I have a question. This is a very nice approach to prepare oligomers. Congratulations on your accomplishment on this approach. Did you find these oligomers or this complex where you have lysin, hemolysin bound beta to be forming oligomers in solution or only in the membrane?
In solution as well. So the membrane, if we add membrane, then we have more oligomers. So on SDS page, I mean, we run it without membrane, but SDS itself is kind of a membrane, is micelle. Yeah, in this case, we can observe if we add more, if we co-incubate with lipid membrane, like lipid vesicle, we are able to see more oligomer formation. So
do you think they're the same type of oligomers, both in solution and in membrane?
Yeah, we only can see heptamer.
Oh, okay, so do they depend on the type of lipids used in the membrane?
Oh, this which we haven't tried. We only use the DOPC and the DOPG, and this is a good question. We can try with different lipids, maybe can change the oligomeric state.
Let's see. There's a question from Shai Rahimipur. Very nice talk. How stable are your oligomers in different conditions?
Yeah, so we in our publication in ongoing tissue, we actually check the stability of oligomers. So they are very stable, and so even after heating, I mean after heating, we see it become the oligomer become further aggregates. So if we stay after kinetics, you can see after the THT fluorescence kinetics, so we can see it it can be very stable after 24 hours at 37 degrees. I think this would be very stable, especially for structure determination and the antibody screening, it will be sufficient.
Did you say that A-beta 1 to 40 does not form oligomers with the hemolysin?
So they can form oligomers. I mean, if we engineer on alpha hemolysin, we always can see oligomer formation, even without a lipid membrane.
There's a question from Christian Grisinger. I'm going to promote him to the panel. He can ask you directly. Christian, go ahead.
Hi, now my question is gone.
Yeah.
So I have to remember now what I was asking. The first question was
in the Q&A, you can read it. If you want to read it, you can read it.
I can read it?
Yeah, the Q&A folder is still there.
Yeah, but I cannot access my, no, I cannot
Okay.
read. It doesn't matter. I think I remember.
Okay.
in the hemolysin A-beta hybrid, when you raise antibodies or nanobodies, most of the protein is, most of the cytosolic part of the protein is the hemolysin. So how do you select then antibodies that really recognize the A-beta?
So we can engineer transmembrane area. For example, we can extend the transmembrane area further with we not only replace the transmembrane area, we can also extend. We can keep the transmembrane area of alpha hemolysin, and then just insert amyloid beta and also alpha synuclein. We are able to see give more like without a lipid environment for antibody screening.
I see.
Did you say that you get these pores also in the absence of membranes?
Yes, we can see it without membrane. It also can form oligomers, only single oligomer, heptamer oligomers.
But
yeah,
the structure must be quite different then, I could imagine, because I mean, if you form an oligomer in a membrane, then at least the outside amino acids have to be very lipophilic, which would not be compatible with solution in water. So that I wonder whether you would then go for the same structure.
Yeah, so we purify the protein with DDM, with detergent, or with detergent. And from the structure determination, we also have DDM, which also is a type of detergent like a micelle model. So, so I mean, indeed this is artificial oligomers. We also want to further confirm it in other in vivo setup. For example, we added this artificial oligomers in a biopsy and example, and let's see how it changes the tissue structure, which now we still collaborating with other group. And meanwhile, we would like to see if we inject this oligomer in mouse, and can we change the pathology of mouse or not? And I mean, there's lots of questions we need to further to confirm it. But I think this is very interesting way to block the ghost, oligomer ghost, which it's quite difficult to catch by other methods.
Could I ask another question, Hans?
Yeah, go ahead. Yeah.
As far as I remember, also truncation variants of A-beta 1 to 42, namely 17 to 42, also form pores. And my question is, in the structure that you showed, that was guiding you in order to make then these oligomers, I think 17 is included in the beta strand. And that's, so the question is, how is that? How does it influence your approach?
So yeah, I mean, we also consider selected the main sequence which can form oligomers or can form pore, like from 17 to 42, and so we in the structure, well, basically this is from Rosetta Zamprine, and the resolution is not sufficient for us right now. We can build a model, but it's not mainly from the simulation fitting into the electron density map. But I cannot see this absolute structure, oligomer structure. It's combined the simulation together with the electron density map. And from our structure, we found mainly the sequence from 17 to 42 can form helping as well.
Okay. And maybe a last question I had, I also remember that zinc ions close A-beta pores. And I was wondering whether, I mean, it could be, of course, that with hemolysin stabilization, it is, I mean, too rigidly fixed in the open conformation. But did you ever look at the reaction of zinc on these pores?
Yes, so the next step, we would like to add zinc and copper. We tried some very pilot experiment. We still need to further explore. This is a very good question. And I mean, apparently we also can screen some compounds or antibody which can block pore. So this is the other purpose we can use I mean, we can screen how the pore amyloid beta pore interaction with ligand or with metal ions.
Okay, thank you very much.
So I
Thank you.
have a question related to Christian's the solution versus membrane oligomers. In solution, the oligomer assembly is such that the exterior should be hydrophilic. In membrane, it should be opposite. So when you screen for antibodies, they're exposed to different surfaces. So there must be a difference in the outcome.
So I have to clarify what just to talk about in solution. Sorry, in solution, actually we had DDM, and which is type of membrane lipidic environment. So I also understand that the in solution and also in membranes, they are quite different condition. So from my understanding, so we for antibody screening, we insert amyloid beta and alpha-synuclein into. It's different from structure determination. We insert amyloid beta and alpha-synuclein into the bottom of transmembrane part, and we again also use DDM as a detergent to stabilize the oligomer assembly, and we thought this is more physiological relevant because assembled helping can permeabilize lipid membrane, and this is one of the main hypotheses. If we purify them in DDM condition, and then we are able to give a more physiological relevant condition. I mean, if we only use like dissolve the oligomer in PBS other without any lipidic environment, indeed it can form oligomers, but this would be not active form conformation of oligomer in lipid membranes. So that we thought the active form would be in lipid membrane environment. So everything we set up with DDM and also with other detergents, which can give more a lipid environment.
So in your synuclein case, where you stabilize the alpha helical structure by the compounds from Andy Hamilton, are these transmembrane orientation? Because I think these compounds stabilize the transmembrane helical structures, right? At least that was the case in IAPP. So do you think your synuclein is transmembrane, and which region of synuclein is helical? Is it still the N-terminus?
Yeah, I think NAC part would be the helical structure, and we added single-stranded DNA, which give more space. Like we use single-stranded DNA as a spacer, the single-stranded DNA can get into alpha hemerycin pore, and NAC terminal would be still on lipid membranes. So yeah, I mean, from single nanopore study, we would not know exactly which residue interact with copper or with zinc. Based on other literature like NMR studies from you guys, we know we can think about which probably interact. So with current data, perhaps not sufficient to figure out which exactly residue from alpha-synuclein interact with metal ions.
Okay, thank you.